be normal during FEMO. We usually have a presentation of one of our. Cancel all of that. This morning, the presentation will be by Eric Dumonteil. Again, we're continuing on a theme of parasitic infections, and particularly in Chagas disease. Eric received his master's degree in endocrinology from the University of Lyon in France, and his PhD in biophysics, in biochemistry and biophysics from University of North Carolina, Chapel Hill. He worked for about, I'm not exactly sure how the connection was made, but worked for almost 8 years, I believe, in Mexico on the disease of leishmaniasis and T. cruzi. Again, a bunch of kinetoplasts, at least is a theme there, as well as work with triatamines in 2 endemic areas of Mexico, as well as you've seen, in Louisiana. He is director of the master's program at Tulane University. And in the Department of Tropical Medicine and Infectious Diseases. He's going to discuss some of, I think, exciting work in terms of development of vaccines for the disease of Chagas. Eric.
Thank you, Ron, for this nice presentation. How do I move? No. No. How do I move the slides?
Forward, backward.
Oh, okay. This is my only slide in Portuguese. So I'm going to be discussing not only vaccine, but also some more of the basic research we've been doing. So it's really a different presentation from everything you've heard in the course, because it's more basic research. But I hope, and I will try to convince you that this basic research, molecular biology, is really important to understand transmission of diseases and how we can control them. So thank you to all the previous speakers from the course and to Claudia, because they've made a lot of the introduction easier for me. So you've seen this transmission cycle for Trypanosoma cruzi. Bugs get infected. When they suck blood from an infected host, then the parasite will multiply in the digestive tract of the bug. And then when they're going to blood feed again on the host, they're going to defecate, deposit the infected feces on the host, and that's how infection can start. And Trypanosoma cruzi can infect a very broad diversity of hosts. Virtually any mammalian species is susceptible to infection. So that includes us, but of our pets, domestic animals, and all kinds of sylvatic mammals. So it's a very complicated transmission cycle, and I'll come back to that in a minute. In terms of the disease, it starts by an acute phase that lasts about a few weeks with no specific signs and symptoms. It's like any flu-like illness, fever, you feel bad. So really, physicians rarely see acute cases, because who goes to the doctor for a little bit feeling bad for a few weeks? Most people don't even go to the doctor. So the only typical signs that this is Chagas disease is that if you look in the blood, you can see multiplying parasite in the blood. Or the Romagna sign, which is this typical biparpidal inflammation of the eyelid when that's a port of entry of and where the bug bites you. That's why they're called kissing bug, by the way. But then patients enter an asymptomatic phase. Apparently, they have controlled the parasite. But they haven't. It's just the parasite is not in the blood anymore. Now it's intracellular and targeting muscle tissue, cardiac muscle, smooth muscle, skeletal muscle. That's why we don't see it anymore, but it's still there. But apparently there's no disease. So who's going to go to the doctor if they don't feel anything? No one. So we're not going to see all those patients. But then a fraction of those patients will start to develop cardiac arrhythmias. Initially, those arrhythmias are benign, but then they're going to be an increasing severity leading to cardiac failure and death. So that's really where the problem is with Chagas disease. This is a severe cardiac disease, and then some patient, as Claudia mentioned, will suffer megaesophagus of megacolon. So why only a fraction of patients develop disease and all the others are fine? We have no idea. And that's a big problem, because you diagnose somebody, you say, okay, that patient is infected with cruzi. What's next? What do you tell the patient? We have no idea who's going to progress and develop severe disease, who's going to stay asymptomatic for the rest of his or her life. And I'll come back to that in a minute. So as you can see from that brief introduction, this is really different from a lot of the diseases that we've been discussing during the course. First, we're not talking about outbreaks, except as Claudia mentioned, those foodborne outbreaks, and there've been a few. But what's the normal ongoing transmission? We have millions of cases in the Americas. Case definition, we've been discussing that in the course. How do you define a case? Most patients are asymptomatic. We don't see the acute phase. We see them when they start to have cardiac problems, when there's clinical disease. Claudia mentioned also, diagnostic test performance. Are we really detecting all infections? What's the sensitivity and the performance of our test? So in some of the previous talk, we heard that, for example, for salmonella, or I forgot some other diseases, we detect like 4, 5, 6% of actual cases of diarrhea, for example. With Chagas, we estimate that less than 1% of actual patients get detected. So we're really seeing only the tip of the iceberg. Are those patients, that 1% or less than 1% that we detect, representative of all infected people? Probably not, but we don't really know. So we're really seeing only a fraction of those patients. This is a chronic disease. Acute diarrhea, COVID, it's really easy to tell if you're sick. You know, it's acute disease. You go to the hospital, to the doctor, because you're really feeling terrible. But what about chronic disease that will take decades to progress? How do you study that? Students don't like that. You say, okay, you start your thesis now, and it's going to take 10 years for you to finish to see how the patient progressed and who got sick and who got severe disease and who stayed asymptomatic. Not very easy to study. Claudia talked a little bit about that. It's not a bacteria, it's not a virus, it's a protozoan. What does that mean? There's a pretty large genome and pretty stable. So again, if we're thinking about genotyping, and Claudia talked in more details about that, so I'm not going to spend much time talking about this. How do you select marker? Protozoan parasite. We have between 15 to 20,000 genes. So it's huge. If we use some of those fingerprinting methods that we can use for bacteria or for virus, we're going to have 1,000 bands in our gel, so we're not going to be seeing anything. So we need to find new methods for those kind of pathogens. And finally, as was mentioned also before, after the acute phase, the parasite is intracellular. So how do you detect it for genotyping? How to do a cardiac biopsy, right? So it's going to be very difficult to identify the parasite in the host in the chronic phase. So these are additional challenges that we have to really study Chagas disease. So what's been done, and you probably all know that, all those of you who work in disease surveillance, vector control. And for the past 50, 60 years, there's been massive indoor residual spraying of pyrethroid type insecticides to kill kissing bugs in people's house. And this map shows you the distribution of Trypanosoma infestans, which is the main vector in South America, and how it's been very nicely controlled by those insecticide campaigns over that 50, 60 years period. And at the same time, the number of infected cases have dramatically decreased from over 30 million back in the 1990s. Now we're around 6, 7, 8 million in the America. So it's looked like this has worked pretty well. But actually, if we looked at the diversity of vectors that we have in the continent, what we've achieved with insecticide spraying has been the very good control near elimination of domestic Rhodnius prolixus in Central America, and the very good control of Trypanosoma infestans we saw on the previous map. But really, all this insecticide spraying hasn't done much against all the other species, and there are multiple reasons for that. The main reason is probably because all those vectors are more sylvatic. So if you spray insecticide in a house, you kill the birds that are in the house at that time, but you have all those sylvatic population that can move again back in the house and reinfest houses. So insecticide spraying isn't working very well with all of those species. And again, once we've removed Trypanosoma infestans of Rhodnius prolixus, then the space is free again for those other species to move in and replace those primary vectors. So coming back to this transmission cycle, what can we do besides insecticide spraying to kill those populations? And to be able to come up with new strategies, new ideas, we need to understand better this transmission cycle, right? What kind of questions would you like to ask? Again, remember, we need always to ask questions. What would be a first question we'd like to understand is this very general transmission cycle? Any suggestion? Now you're asleep. Well, first of all, those bugs, what do they like to feed on? Do they have any feeding preference? Do they prefer to feed on dogs, on us, on chicken? Because then that would give us ideas who's most at risk, right? What else would we like to know? Of all those potential hosts, yes, they're all susceptible, but some might be more susceptible than others. They can be what we call the super spreaders, those that really maintain a very active transmission cycle. So maybe if we identify the host that's really the super spreader in that cycle, remove it, then maybe we can interrupt transmission. So what's the susceptibility of all those different mammalian species? And what Claudia mentioned, we have multiple strains of parasites and lineages. Are they all circulating the same way? Maybe we find that some hosts concentrate some sort of lineage A of the parasite, another host, they're more susceptible to lineage B. So we actually have subcycles within that general cycle. So really, we need to understand that textbook cycle in much greater detail if we want to do something else and improve vector control. So that's where molecular biology can become helpful. And we heard in the course a little bit about metagenomics. And really the idea is to do detective work to understand better the biology of the vector. So we extract DNA from the vector, and then we're going to be, by PCR, amplifying different markers. We can use a marker for species identification and some population genetic of the bug to see how the bug population is moving around and identify which species of Trypanosoma we have. In the same DNA sample, we can identify if there is any vertebrate blood that will indicate what was the feeding profile of that bug. We can identify if the bug is infected with the parasite and do some genotyping to see what strain of parasite is present. And again, the parasite is in the digestive tract of the bug. So it's along all the bacterial microbiota that's there. But what's going to happen between all those bacteria and the parasite? There might be some interaction. They might exchange nutrients, or they may inhibit one another. What's happening? And again, the nice thing about those approaches, these are the 4 markers that we've been using, but you can use more markers. You can look for all kinds of things. You could look for viruses at the same time, fungus, anything. And you sequence all these, and you're going to be able to tell for each individual bug what it's been feeding on, if it's infected or not, what bacteria are there, and what species of bugs we're dealing with. So this is an example of a study we did in southern Mexico with Trypanosoma immediata, the vector there. And these are the feeding sources that we identified with these methods. It's feeding on human, dogs, and really all kinds of animals. So the first message is we don't really see a very strong preference for anything. It looks like it can be feeding on pretty much anything. And both male and female bugs have really the same feeding profile. But that's not the key information. Because of the sensitivity of next generation sequencing, we can actually reconstruct the feeding history of the bug, not only the last meal, but we detect on average, these are all individual bugs, and the colors represent different host species. So on average, we can identify almost 5 different species in the stomach of a single bug. That's its recent feeding history. And looking at bugs in the lab, we know that it's about 1 month of feeding history of the bug. So that gives us very precise information. How often do they feed? How often do they switch host? And that you can look at just the diversity of colors. You can tell that they change host pretty frequently. And they really jump from anything. Look at that bug here. It has blood from human, dog, mouse, rat, cow, bat, squirrel. If that bug is infected, it means they can really move parasite around across all those species. Obviously, there's not going to be 100% transmission each time it feeds, but statistically, the parasite might be able to move across all those species. But now we can use this information to reconstruct much more detailed transmission cycle. It's a complicated graph, but it summarizes the transmission cycle. So the size of each host is proportional to the feeding frequency that we've seen. So we can see that they are feeding on human, on dogs, then on some other animals. But now we can connect those based on how frequently we find 2 species in the same blood meal in the same bug, because that indicates that's a high probability of parasites moving, for example, between human and dogs. So what might be the host most important source of parasite for human infection in that particular setting? Look at this thick line here, dogs. On the other hand, I don't know the name of that animal, but look at this thin line. It's probably not a very good source of parasite. But still, you can see that many different hosts can be sources of parasite for humans. So there's not a unique super spreader that we identify. So it's going to be complicated, really, to interrupt that transmission cycle because it's so complex. That's southern Mexico. We've been doing similar studies in other settings, and I don't really have time to describe all those settings, but the idea is that they correspond to completely different ecological scenario and transmission intensity. So we have sites in the US where it's more like spillover transmission. We have a few human cases here and there. Here in the Yucatan, we have about seroprevalence in the human population is between 1 to 5%, or a bit more in Veracruz, where it's up to 16% of the population is infected, so there's moderate transmission to human. We have some hyperendemic area in Colombia with almost 50% of some indigenous community are infected. Or in Ecuador, where Trypanosoma immediata is actually an introduced species. It was introduced in the colonial time by ships from Central America to Ecuador. So the idea of comparing transmission cycle across all those different eco-epidemiological conditions is to see what do they have in common, what's unique to those places that can give us an idea of how to mess up and interrupt this transmission and decrease risk to humans. So these are some of the different cycles that we obtained. This is Veracruz in Mexico. This is in Belize in Central America. This is in Colombia. This is the same vector, Trypanosoma immediata, same species, different eco-epidemiological condition. Do they like to feed on anything special? No. Whatever crosses their path that day, they don't care. They're going to try to feed. So again, it's going to be very difficult to interfere with that transmission cycle. But there is something in common. Look at the dogs. Dogs are an important feeding source. And when a bug feeds on a dog, most likely it has also human blood. We don't know in which order it's feeding, but there are a lot of interactions. And that's really nice because you know what? Epidemiological studies without all that molecular biology had shown before a statistical association between having dogs in your house and increased risk for humans. But now we have a mechanistic explanation for that statistical association from epidemiological study. And using that mechanical information, now we can build mathematical model using those feeding frequencies, this co-association between hosts, to have a mechanistic model that describes transmission. And I won't go into the detail of that model, but basically we have an infected host that can be in contact with an infected vector and become infected, and uninfected vectors can become infected if they feed on an infected host. But now we can parameterize this model very precisely with our information from those field studies or that molecular biology to really reproduce transmission and ask questions that what's the contribution of each different species to transmission? And we can start thinking about new control measures. For example, what if we put insecticide repulsive collars on dogs to prevent dogs to become infected? Could that work? Could that interfere parasite transmission to dogs and to humans as a consequence? I won't get into really much of the details. Obviously, we don't really know how those collars would work, but this is a sensitivity analysis in which we varied the proportion of dogs with collars from 0 to 100%. How effective are those collars? We don't know, but we vary again between 0 efficacy to 100% efficacy, stopping completely triatomine feeding on them. And we can see, well, you know, we don't really need an extremely efficacious collars on all the dogs to get some effect. Just about 50% efficacy of the collar on 50% of the dogs, we can reduce significantly infection in the dogs. And I don't have time to show you, but that will also translate in reduction of human infection. So the message is, by understanding better this transmission cycle, we can start thinking about something else than insecticide spraying. I mentioned the bacteria in the digestive tract of the bug. So if we're doing that with that approach, we can identify all those different bacteria in the digestive tract. But look here, what's interesting is that if we compare uninfected and infected bugs, they have different bacteria. So that means there's probably interactions between the bacteria and the parasite. And not only that, Claudia mentioned the different lineages of the parasite. Different lineages of the parasite present in the bug, and we have different bacteria. So that speaks of very specific interactions, and not all strains of the parasite will interact in the same way with the bacteria. Some bacteria may favor the parasite, some bacteria may inhibit the parasite. We still don't know, but it seems that there are very tight interactions. And what's those bacteria doing for the bug? That microbiota is important for the bug. In that study, we compared the microbiota of nymphs during development with adult bugs. Very different microbiota composition. And if those nymphs don't acquire those bacteria, the development stops and they die. So they need to acquire the right bacteria. Again, we don't know what those bacteria are doing for the bug. They're probably digesting some products from the blood, providing some key nutrients, but they're doing something important for the bug. And if we compare from different habitats, and this is in Trypanosoma sanguisuga, our vector in the US, comparing the microbiota of bugs from Louisiana to Illinois, they have completely different bacteria. You do the statistical analysis, it's completely different. But if you look at the metabolic properties of that microbiota, which means what, you don't have to read all those chemicals. These are all the carbon sources that those bacteria can metabolize. And these are all the fermentation products that those bacteria can produce. Even though the bacteria are different, there's a very tight correlation in their metabolite profile. So the microbiota is different, but its metabolic function is conserved. So that tells us those metabolic functions are probably very important. It doesn't matter so much which bacteria is going to do the role, as long as the metabolic reaction is there. So again, that tells us that the microbiota is important. And if it's important for the bug, maybe we can mess up with that and make the bug unhappy and find better tools than insecticide spraying. So really now we know that there are very tight interactions between the parasite and parasite strains, the bacteria from the gut microbiota that can affect parasite development, transmission, and bug growth. So maybe that's maybe the next generation of vector control tool. Interfere with the microbiota to make bug refractory to T. cruzi infection, to slow the growth of the vector. And this is not a new idea, but over 20 years ago, colleagues at the CDC proposed to use bacteria to make bugs refractory to T. cruzi infection. But 20 years ago, they didn't have those molecular tools to really leverage that. But now we have. So if we understand really much better those interactions, maybe we'll come up with something better than insecticide spraying. Switching to subject, Claudia mentioned the diversity of parasite and that we are now 7 lineages of Trypanosoma cruzi. They're probably they probably have very important biological difference. And by the way, for those of you who know better Leishmania, the amount of genetic diversity within those lineages of T. cruzi is similar to the genetic diversity between Leishmania species. And you know that with Leishmania, if you're infected with Leishmania infantum, you get a very different clinical profile than, for example, Leishmania brasiliensis. So what's happening with T. cruzi and those different lineages? And Claudia mentioned that using molecular method, we started discovering something that we think is important. These are those co-infections. We don't have just one T. cruzi strain in a host. And these are examples from some of the studies that Claudia showed you. In rodents, we found all those different circles indicate a different genotype in a single mouse. Look at those dogs infected with different genotype of TC1, TC2, and TC6. That one has TC1, TC4, TC2, TC6. So really, a large proportion of infection with T. cruzi involve parasites from multiple genotypes or lineages. And that holds for hosts, but also in vectors. So how does that work in real life? Are those infections simultaneous or sequential, that they get first one lineages, then another one later on? We don't really know. What are the implications for parasite selection, amplification, and those transmission cycle? We don't know. Claudia talked a little bit about that for diagnostics. How it's going to affect diagnostic? Probably a lot. Because if your test is designed to detect specific lineage, it's going to miss the other one. And she showed example of that. And what about for the pathogenesis and disease progression? And if we go back to what I mentioned at the beginning, why only some patients progress to severe disease and others remain asymptomatic? Could infection from multiple lineages be an explanation for the pathology? So to try to answer that question, we turn to those monkeys that they have at the Tulane Primate Center. The Tulane Primate Center is a huge facility from Tulane. It's about an hour away from New Orleans. They have over 4,000 monkeys It's a breeding colony because they provide monkeys for all research centers in the country that do animal research. But those monkeys are housed outside. So no matter how well maintained they are, about 1% of their colony is naturally infected. For them, it's a problem because they can't use those animals for medical research. But for us, it's a gold mine, right? Because these are captive animals. So we've been using those animals to follow disease progression. And now we've been following them for over 3 years to look at disease progression. And we're also using them for vaccine and drug development. So the first thing we start looking is at their level of parasite in the blood. And so over a 2 to 3 year period of follow-up, each individual line is an individual monkey over time. And we could see immediately two groups. One that we call progressors, in which there is a slow increase over time of level of parasite in the blood. And those that we call controllers, in which it's stable or even decreasing over time. It doesn't go away, but it seems to be decreasing. And when we looked at cardiac disease, this is one of the monkeys that actually died at the beginning of our study, which had a huge cardiomegaly from cruzi infection. This is the normal heart. And this is how enlarged the heart of that monkey was. But when we looked at electrocardiographic recordings over time of those monkeys, we found that those that we call progressors, we can see change in their electrocardiographic parameters over a 6 month period. So they have progressive cardiac disease as well. Whereas those that we call controller, they remain asymptomatic, no change over time. So really what we see at the parasitological level, we see it reflected in cardiac function. So then we ask the key question, okay, what are they infected with? And we went back to our genotyping. And we identified TC1, TC2, TC5, TC6, TC4. But with our advanced genotyping method based on next generation sequencing, we can detect those co-infections and not only detect them, but quantify the relative proportion of each lineage in each monkey. And this is what's described in that figure. So they all have mixtures of parasite, but in different proportions. But if you look at that, really progressor monkeys, they're mostly infected with 1 or 2 parasite lineages, the majority. Whereas the controllers, they're infected with 4 or 5 lineages at the same time. For us, that was counterintuitive because we thought that maybe if they're infected with high mixture of parasites, they're going to have more severe disease. But it's the other way around. If they're infected with many lineages, they seem to be controlling the parasite better. And really, if we looked at the number of haplotypes, there's a very significant correlation. The more diverse parasite they have, the lower the parasite burden. But if they have just few parasite lineages or just one clone, the parasite proliferates. That's interesting. So then we did a follow-up of those monkeys over time. And we found 2 general patterns. When there's just 1 or 2 lineage of the parasite, we see things like this. Pretty stable over time. A comfortable infection, the parasite is happy, it's maintained over time. But when we have multiple lineages, we see things like this. What does it look like? It looks like the parasite, they're all fighting each other, trying to become the dominant parasite in the host. So we start with a lineage, then another lineage is able to repress the other strain and be the dominant parasite, but that doesn't last long, and then another one come up, and it's really looked at as competition among the parasite strain. And maybe that's why the infection is not so successful from the parasite point of view, because they're really competing against one another. But with those data, we were able to calculate the actual multiplication rate of the parasite in the host. And we can see that definitely the parasite multiplication rate in that host depend on the lineage. So some lineage are able to better proliferate in that particular host than other lineages. But even that really depends on what they're surrounding, because when there are a high diversity of parasites, they're not able to proliferate that much. But when there's little diversity, they can proliferate more. So it really looks like that this multiplicity of infection is counterproductive for the parasite and beneficial for the host. So we came up with a new explanation, potential explanation and hypothesis for disease progression. Maybe it's all determined from the onset of infection. And if at the beginning you're infected with a very small diversity of parasites, you can have a severe acute phase. There won't be any competition because it's just a few clones of the parasite. They're going to be able to proliferate and lead you to severe disease. Whereas if you're infected at the beginning with a high diversity of parasite strains, there's going to be competition among those parasites and not so much disease progression. And that would be really important because maybe now we could have a good tool to predict patient progression. Who's going to be progressing to severe disease, who's not going to be progressing to severe disease. And to better understand the mechanism under this, we started doing transcriptomic analysis, looking at gene expression of the host to see how the host is responding to those different parasite strains. And just looking at this principal component analysis, monkeys that control the parasite have a different gene expression profile than monkey with progressive disease. And we could relate that to differences in the immune response. Some of the immune genes from those immune cells in the blood. Some of the genes are common to both groups, but then progressors and controllers have unique up and down regulated genes associated with the immune response. So the host is responding differently depending on what mixture of strain is present. But not only that, but when we looked at the gene expression profile between our controller and our progressor, we actually see three groups of different immune response. What's going on in each of those groups? These are the genotypes they're infected with. And you can immediately see that the host response really depends on what strain mixtures is infecting that host. Those are mostly infected with TC1. Those are mostly infected with TC4. Those have the greatest diversity of parasites in our controller monkeys. That also include TC5, TC6, and TC2. So really the host response in terms of gene expression, immune response, matches parasite diversity that's infecting those hosts. And we do find differences in the immune response of these are our three groups of gene expression response. There is difference in cytokine and CD molecules of the immune response. And we can identify new biomarkers. These are different molecules that we can measure in the blood that help us classify the monkeys, depending on what's their host response and what's the genotype of parasite included infecting those monkeys, so that we can better predict disease progression. So really what we know so far is that there's a large proportion of infection that include parasites from multiple genotypes. And those with high parasite diversity are associated with lower parasite burden and limited disease progression. So host response is modulated by parasite diversity, and this is a key factor driving disease progression. But now we're interested in using those macaques for testing drugs and vaccines. As you know, we only have two drugs to treat Chagas disease patients, nifurtimox and benznidazole. Both have limited efficacy in advanced stages of the disease and side effect, and really few patients actually get treated. So over now 20 years ago, one of an undergrad student in the lab, Norma, started testing a therapeutic vaccine, and she did a very simple experiment. She took mice, infected them with the parasite, then give a vaccine, a therapeutic vaccine, to boost the immune system and see what happened. So in control mice that don't receive any vaccine treatment, the parasite proliferates in the blood, and all the mice die within 40, 50 days. It's a lethal infection. But if you give them just 2 doses of a vaccine that will boost the immune system, you reduce the proliferation of the parasite in the blood. This is just counting parasite in the blood during the acute phase. And that's a vaccine encoding this antigen, parasite protein TSA1, and this is another parasite protein, TC24. So those vaccinated animals control the parasite, and most survive this acute lethal infection. So this was really the first demonstration that a therapeutic vaccine can control infection in a mouse. That was 20 years ago. Since then, you don't have to read all this table. The only point I want to make that us and many other groups throughout the America using very different types of vaccines, from DNA vaccine, peptide vaccine, recombinant protein, viral vectors, have found similar results. And using all different kinds of parasite antigen, not only those 2 that I mentioned, I've been able to reproduce those results in mouse model. So this is really strong evidence that a vaccine might be feasible for Chagas disease. So because of this evidence, we were fortunate enough to integrate a public-private consortium, because you cannot do vaccine development on your own. So we partnered with all those institutions, including Peter Hotez and Baylor College of Medicine, institutions in Mexico and at Tulane, with really the goal to promote and vaccine development against neglected diseases. Because as you know, there's no interest from the pharmaceutical industry to develop vaccines for something like Chagas, because there's no money to be made. And that's what drives them. Can they make money? That's why they were interested in the COVID vaccine, because they make themselves rich, not because they wanted to save the world, right? So the first thing we did in our consortium was, well, what exactly would we like to have as a vaccine? So we defined this product profile, and we would like the vaccine that can stop or at least delay cardiac manifestation, because this is really what's causing problems to patients. When they start having cardiac manifestation and they cannot work, they need heart transplants. And that's really important, because we're not really targeting to get rid of the parasite. Do we really need to get people rid of the parasite? 70% are going to be asymptomatic no matter what, even though they may carry the parasite for the rest of their life, right? So that's an acceptable target, to just prevent cardiac manifestation. We want that vaccine to be able to work in adults and children, as few doses as possible, so that it's easy to administer, and an efficacy of at least 80% or a delay of at least 10 years of those cardiac manifestations. So to accelerate vaccine development is a very complex process. It's not only doing preclinical evaluation and then clinical evaluation of vaccine. That's an important component. But you need to be able to produce your vaccine at industrial scale. You need to have processes for formulation, optimization, quality control, stability, et cetera, do technology transfer to a vaccine manufacturer, all the regulatory aspects with FDA in the US, Fiocruz is in Mexico. I don't know what's the name of the regulatory agency here in Brazil, but you need to go through all those. Okay, you need to go through all those regulatory process. You need to do cost-benefit analysis so that you can have investors, etc. Usually this is done sequentially, and that's why it takes forever to have a vaccine. But we've been trying to do everything in parallel at the same time. And the idea of doing everything at the same time is that if we detect something wrong at any one of those steps, we can try to address it immediately and move forward. So I'm going to concentrate on preclinical studies because this is really the part I do. So first, we showed that our new formulation of our recombinant antigen do work. This is in mice. So this is a mice that was vaccinated with the TC24 vaccine. This is unvaccinated control. You can see all those amastigot nests. This is the intracellular form of the parasite in the heart of those mice. With the vaccine, there's still some parasites, but that heart tissue looks much healthier than this one. Same thing with the other antigen. So our antigen work in mice. But as I mentioned, decreasing parasite function, parasite load is good, but really we want to do cardiac function. This is echography in a mouse. This is a normal mouse, normal beating heart. You don't have to be cardiologist to see that something is wrong there. It's really not beating correctly. And that's what we would like to prevent with our vaccine. So now we can do study in mouse to try to prevent those cardiac alterations and measure the efficacy of our vaccine on cardiac function. I won't see you more mouse studies because really you can cure everything in a mouse model. We can cure cancer, we can cure HIV, we can cure everything. So we're interested in monkeys. And again, we worked at the primate center looking at, and this was with an experimental infection with T. cruzi. So we infected those monkeys. And then we gave 3 doses of our therapeutic vaccine and look at cardiac function. We also looked at a lot of other parameters, but I'm just going to focus on cardiac function. So we do EKGs and measure all those different intervals on the EKG. So 3 months, there's still no difference between our control and vaccinated monkeys because there are still no cardiac alteration after 3 months. It's barely the end of the acute phase. But after 7 months of infection, all control animals that didn't receive the vaccine, they all start presenting cardiac alteration. But those who received the vaccine, only 1 presented cardiac alteration, the others remain completely normal. So this is the first evidence that we can prevent the development of cardiac alterations in monkeys with a therapeutic vaccine. So now we've done also immunogenicity studies. With our latest formulation of the vaccine, so in those studies, we just did on naive monkeys. They received 3 doses of the vaccine, and we looked at the immune response. We have very high antibody levels against both antigens after each of the different doses. And interestingly, this is a Western blot. Those antibodies recognize the native protein in the parasite lysate. And I forgot to mention that those 2 antigens are highly conserved in a lot of parasite strains. So we will hope that that vaccine would work against a broad diversity of strains. We've also been doing molecular analysis of the complete T-cell receptor repertoire of those monkeys. And again, those network, each little circle is a different T-cell receptor. So really capturing the whole diversity of the T-cells of those monkeys after each of the vaccine dose. And you can easily see that some particular T-cell start to proliferate. These are the T-cells that are responding to our vaccine antigens. And now after each dose of the vaccine, some of those very specific T-cells responding to the antigen are proliferating. So now we're just getting started a non-inferiority trial to really compare the efficacy of this vaccine in macaques naturally infected using those macaques that I described earlier, in which we have a very clear description of what strain they're infected with, how long they've been infected. And the idea is to compare the vaccine to conventional benzimidazole treatment. That's why it's a non-inferiority trial. So it's really designed the same way a clinical trial of that vaccine would be designed. Because obviously, ethically, now we can have a placebo group of Chagasic patients that won't receive any treatment. They need to have the benzimidazole treatment. And the idea is to show that the vaccine can be equivalent to the benzimidazole. These are the different groups that are involved in this project at Tulane, at the Primate Center, Baylor and Cinvestav in Mexico, which are our biotechnologist colleagues who are in charge of all the biotechnology to produce the antigen. And really, now we've genotyped many more monkeys. We know exactly what strains they're infected with, and the idea is to follow them for 1 year after therapy, either with benzimidazole or the vaccine. And the outcomes we will be measuring are obviously parasite burden, cardiac function, but we will also look at a number of biomarkers of disease progression and immune response, to try to identify molecules that are easy to measure, because obviously what I'm showing you, it's a lot of molecular biology and fancy technique that we can do in research, but we'd like to have something that can be applied in a clinical setting. When you have a patient, let's measure something easy to detect in the blood so that we can predict disease progression. So we can use all that cohort of monkeys to identify biomarkers of treatment efficacy and disease progression. So we still have a long road to go for the vaccine. We're still here doing preclinical studies, looking at efficacy in non-human primates. I don't have time to mention, but we also have a field study in dogs of our therapeutic vaccine, trying to decrease infection in dogs, and that's a field study we have ongoing in Mexico. Hopefully the results of those studies will be satisfactory so that we can start clinical trial of our vaccine in human, phase 1, phase 2, phase 3. But you can see that we still have a long way to go to have a vaccine for Chagas disease. But I think this would be a fantastic tool to have in the future. And we're really excited about being part of this effort. So I'd like to thank colleagues in the lab, Wei Hong, our lab manager, Hans, who've done a lot of transcriptomy, Curly, Claudia, obviously, and Pierre, our Chagas colleague at Tulane, the folks at the Primate Center, a large number of collaborators for field studies throughout the US and in the endemic countries, in Mexico, in Colombia, in Yucatan, and happy to take any questions.
We have about 6 minutes for some questions. I think both for Claudia and for Eric. Yes. Hi.
Oh, just a second.
Vou perguntar em português, tá? Primeira, a questão da vacina. Não seria necessário realizar um inquérito na população para poder identificar se realmente seria necessário vacinar a população, nesse caso. O segundo ponto, se a doença provoca problema, arritmia, problema no coração, uma vez que eu tomo a vacina, eu não poderia, pensando na Covid, eu não poderia ter como consequência encadear um problema cardíaco após a vacina. E a última, quando tiver surto de Chagas alimentar, é possível fazer um inquérito aqui no Brasil para testar a população, para ver a prevalência da doença de Chagas nesse local.
Okay. So the first question was about vaccinating the population. We're really not thinking about vaccinating the population. This is a therapeutic vaccine, so that will be only for patients. So infected persons, instead of giving them benznidazole for treatment, or in combination with benznidazole, you could give them both. To improve treatment efficacy, you know that with benznidazole, there are severe side effects. A lot of people don't complete the treatment because of the side effects, so there's low compliance, and that's an issue. So maybe with the vaccine, we can have greater adherence if it's just a couple of doses of the vaccine, and then we get the same therapeutic effect. So we're not thinking about vaccinating the whole population, but just patients. Your second question is, if I understood correctly, is like about side effects of the vaccine that would increase pathogenesis in some way. Is that correct? Yes. That's certainly an important concern. And in all the preclinical work that we've done in mouse model, in dogs, and now in monkeys, that's something we're watching very carefully, because we know that a lot of Chagas disease, the disease is because of some imbalance between the immune system. There's inflammation of the heart, fibrosis. So if you have the wrong immune response, you may increase pathology. We've never seen that in the animal model, but we're really careful about watching that to make sure that that would not happen.
For the last question, Claudia,
Yeah.
a question about surveillance in outbreaks for Chagas types in like foodborne outbreaks in Brazil. What are possibilities?
Thanks for your question, Ron. Yes, it's important to do that in those outbreaks. We have seen some relation with the different genotypes associated with the severity of the acute acute phase in an oral transmission outbreak. Yeah, and actually in Colombia we saw in the south that we have one different genotype is lethal compared with the north part that we have another genotype that we can at least treat the patient. So it's important to have that, and it is possible. We need to do that.
Albert,
Yeah, I think just first comment is thank you, Claudia and Eric. Really great presentation, and Eric, really very exciting work on the vaccine, the therapeutic vaccines. So just a couple quick questions. One is maybe following up, what would be the target population for the therapeutic vaccine? Would it be people before they, maybe it's a little bit related from Harvard, would it be asymptomatic people, or would you give it to, the timing, moving the natural history? The second question would be just your thoughts on the correlative protection, whether it's really going to be antibodies or speaker here or anything. And then the third one, so it's a little bit more extended. So this study may be a very difficult clinical study, clinical trial to do, because of the progression of the disease. And if the follow-up is not long enough, you may only see like the tip of the iceberg, right? The early progression rather than the late progression. So maybe your thoughts on, what would be good, how important would be, and if you put your target product profile, you kind of put really the high bar, right, of a clinical, that might be like dying on the first battle. So were you thinking, and you had really elegant macaque studies, and so could you do just parasitemia? Could you kind of stage this, because it's going to be a really challenging clinical study. But anyway, great work.
No, definitely working with a chronic disease that takes years to progress, it's a challenge. There's no really easy answer to that. We will need to have a long clinical trial. It's not like COVID, in which clinical trial lasted 3 months and then they had a very good answer on the efficacy of the vaccine. So that's impossible with Chagas. We're going to have long clinical trial. But now we have the experience. There have been a number of clinical trials for drugs that can really be informative for us. You do need long-term follow-up of the patients, but it's feasible. But it's feasible within 4 or 5 years of follow-up, which is, I think, the benefit trial was the longest trial. So that's still early progression. And what about late progression? But only time will tell.
One last question.
Thank you. Parabéns para los dos. Felicitaciones. Buena presentación. Maybe I will ask it in English. I have 2 questions, very simple. Would you both expand on, pregnancy, women pregnant, what's the next, because it's so difficult, especially in our countries, to manage the disease. And the other obvious question, coming from tuberculosis, we have this lineage as well. well, and we learned, different ethnicity has better fitness, the bacteria. What's gonna be, as you guys going to study, this interaction, like the fitness of this parasite according to the ethnicity, because it's around the world.
I have the time to show you, but our next time for, our next step for congenital, we need to make sure what are the haplotypes that are associated with transmission. So if we can detect that, we can give that information for treatment, for diagnosis, no? So we can know before with using diagnostic specific tests, if that woman can transmit to the baby, because we know that the 5% of the women can transmit. So that's from my side. So yes, a lot of work to do, and I'm doing that right now.
They have transmission, congenital transmission in monkeys.
Yeah.
That's how they want.
Yeah, there is also congenital transmission in other mammalian species, including monkeys. But with congenital transmission, the rate is low. It's about 5% of infected mothers who will transmit to the newborn. So you need to identify those, and you need really large cohort, which is why, for example, the study that Claudia mentioned, we did in Mexico, Honduras, and Argentina, we enrolled 30,000 women to get like few hundred that were infected, and then even fewer cases of congenital transmission. But what we've been observing recently is congenital transmission is one aspect, but just being born from a mother infected with T. cruzi without congenital transmission, you can have low birth weight and possible issues later in life. And we're trying to address that right now. We identified some epigenetic changes in newborns without congenital infection, but just being exposed to the maternal infection may have issues. And that would really be a serious problem, because there are many babies that are born without congenital infection and say, oh, no congenital infection, they're fine, and they're never followed because we think they're healthy. And that may not be the case.
We have one last question.
More forward.
First of all, congratulate to Claudia and Eric. Very nice presentation. I had some just information, just never have a question. In this place that we are now, for us, Professor Sonia Andrade described the biodem. Here, this area here. It's number one. The number two, my colleague in Belo Horizonte, who was leader by Sérgio Danilo Pena, he looked in autopsy material, and he found that lineage in the gut was different than lineage that he found in the heart a long time ago. The third thing was observation from Professor Sultan Wright, was from here, where he kept dogs here, infected dogs, and the dogs get infected, recover a little bit, they go for the indeterminate phase. Long period. Then he challenged with low dose of cyclophosphamide, and they had an explosion. It was not only in the parasites, but in the disease.
Yes.
That's in relation to the microbiome, I would like to ask you, you are doing for bacteria, but did you like for virus? And in relation to the different parasites, one thing that regulate is, in general, those parasites release the surface molecules. And this they are seen as the exosomes. And those molecules regulate the immune system, and this probably displays something.
No, these are fantastic points. For the microbiome, we've only started looking at bacteria because it's easier. But definitely we should look at viruses, we should look at fungi to really have a good description of everything what's there in the gut of the tryptomine, because there might be some other things besides bacteria.
Very good. Thank you all for coming. We now have,
Deixa eu contar uma historiazinha rápida. Muito importante. É café agora. Cláudia mostrou, vou falar em português para ficar bem claro. Cláudia mostrou o sinal de romântico, não é isso? E é uma coisa que o médico tem que levar em consideração se você está em um local que é muito raro você ver. Aqui nesse espaço, tinha o laboratório da professora Sônia. Eu era estudante de mestrado à época. Isso provavelmente em 1983. E eu era uma pessoa muito agitada e brincava muito com todos. Então ela disse, cuidado com o Mittermeier. E eu era um estresse muito grande, porque trabalhava com cepas colombianas, cepas de São Felipe, todas as cepas. E eu morava numa república aqui em São Paulo. Bom, certo, enquanto eu estava fazendo o meu mestrado e fazendo estágio no laboratório dela, captando animais, um dia eu estava na minha república, morando com vários parentes, irmãos, e no meio da noite, lá para umas 2 horas da manhã, não sei porquê, eu acordei com um dos olhos inchado. Eu tive um edema clássico, unilateral e palpebral. Eu entrei em pânico, eu sou médico, eu sou patologista, fiz várias autópsias, né? E eu disse, eu me infectei. Então, levantei, falei para o meu amigo do lado, assim, meu primo, disse, olha, rapaz, eu acho que eu me ferrei. Eu me infectei lá no laboratório. Mas eu vou ver, não comente com ninguém. Aí eu fui no banheiro, para olhar no espelho, lavar o rosto. Enquanto eu fui, ele bateu em todas as portas, quando eu vi, estava todos acordados. Olha, ele Mittermeier se ferrou. Olha, tá vendo? E aí eu entrei em pânico. Às 5 horas da manhã, eu saí correndo, né, e fui para a porta do Hospital das Clínicas da Universidade de Tulane. O meu olho um pouco fechado. O primeiro cara que eu encontro era um oftalmologista, o professor Roberto Marbach, de Messi, que trabalhava. Aí ele olhou para mim e disse, professor Marbach, eu queria que o senhor me examine. Aí ele olhou para mim e disse, mas um Marte na ciência, mas um Marte na ciência. Aí o meu coração disparou. Aí ele esperava o elevador para subir para a enfermaria, para o lugar onde ele tinha a clínica que ele respondeu os pacientes. Eu disse, professor, vamos andando pela escada. Não, vamos esperar. Professor, vamos andando. Eu queria que ele me examinasse. Quando eu chego na porta da enfermaria, ele disse, você fica aqui fora, você me espera. Eu vou lhe ver aqui nessa sala. Eu disse, professor, mas não tem problema, eu não quero que você infecte meus pacientes. Eu disse, professor, mas não tem barbeiro, não tem nada. Aí ele, fica aí, eu vou voltar. E aí meu coração faltou sair pela boca. Quando ele volta, ele disse, Mittermeier, você está com conjuntivite. Aí quando ele olhou para o lado, eu meti o dedo no olho, passei no outro olho. No outro dia, amanheci com 2 olhos inchados.
Mas eu que o senhor estava um conjuntivite por uma série gonorreia.
meu amigo, não? Agora eu quero que Eric e Cláudia botem no bar a Bahia no próximo curso. Eu, Gilmar, então vamos por encerrada essa reunião de lei. Muito obrigado.